And Professor Bagnato will talk to us today about the control of resistant pneumonia using extracorporeal illumination.
Okay, thank you very much. I want first to acknowledge the efforts that you all doing for us to have activity going on in the middle of so many problems around the world. And of course, we see a large number of people participating, which means that besides the big situation, the bad situation, people are very interested in still keep going with their goals in using photodynamic, photodiagnosis for helping the whole situation that is going to be established. It is now and will be established after all, right? We have to prepare ourselves for the after storm, and we know that it will be hard. And I want to say that the talk of Nick Stone was wonderful, and I appreciate keeping, and I hope all the audience enjoy. Okay, I will be tell you a little bit about PDT for controlling infections in the respiratory tract. I changed a little bit my title to be a little more general than just speak about one of the topics. And since I don't have a full feedback, I hope that if something goes wrong, you immediately warn me that something is not going well. Okay, so I am in the University of São Paulo here in Brazil. We are in a big mess at the moment, as many other parts of the world, but I hope we can get out of this and continue our studies on this photodynamic therapy that we are doing for many decades already. Okay, I have a center here in Brazil that we do many things. You know, my main work was originally in atomic physics, so I am an atomic physicist. But we see that everything we do is related to atoms and molecules, so I consider myself in the right field. We do biophotonics and nanoplasmonics, and that's why it was very much interesting for me, the surface enhancement realm, and I think this will be a great field. And I'm putting here the homepage of my group because I normally don't list all the publications that we have, but you can go to our homepages and find a whole list of publications. Everything that I'm gonna tell you, most of it is already published in journals that we have around. So the center that I coordinate is big. You can see how many people we have, and different laboratories doing different things. You see everybody hanging in the top of a Volkswagen old Kombi, one of those wagons, and they are not in biophotonics, they are in atomic physics. But all the people in white, they are actually related to clinical work. And I will be very much speaking more about some clinical things we're doing, and or we are towards doing those clinical applications, especially in the respiratory tract. As you know, we are facing a big problem. We were facing a big problem before with the resistant bacteria, especially the resistant pneumonia to antibiotics. And that's one of the main causes of people in the intensive care units around the world. And this is the target that we are trying to reach with the photodynamic therapy. Okay, now I have a lot of people, I didn't say the name of all of them, but for the things I will be speaking to you with is the pulmonary disinfection. We have a team here in Brazil that I'm placing here for you to see. And we have a very good collaboration with Toronto in Canada. And those are on the clinical side, we have Marcelo Cipel, and on the production of nano emulsions for deliver well the drug, we have a Professor Gang laboratory, and we have students working many things. Another thing that I'll be telling you is about the disinfection of the endotracheal tube during the intubation of patients, which is common now, right? and the pharyngeal tonsillitis, which are the infections of the throat. And we are targeting those infections as well. And for that part, we have a good collaboration with the University of Coimbra, Mariette Pereira, and including Luis, which is the president of our International Photodynamic Association. Okay, so in the biophotonics part of our lab, we are mostly focused in photodynamic, sonodynamic now with ultrasound, photodiagnosis, and we're trying to go from, you know, basic principles from cells, from individual microorganisms to experimental models to clinics. So we're trying to do that route for many of the projects we're doing. And I know it's not the main topic of our talk, but I want to say that we are very serious about skin cancer because Brazil has at least 300,000 new cases of cancer every year. And skin is a big issue here. And we're having a wonderful program, maybe one of the largest multi-center clinical trial for skin cancer, including some countries in Latin America as well, and even some partnership in Europe. And with this, we have been doing a wonderful work, and I'm showing to you because we want to take the same track for the other type of research we're doing. And here we develop the devices. We are connected with companies that can make what we do really available for society. And this is the skin cancer part that we are doing with, maybe it's the only protocol with real-time investigation by fluorescence to really optimize the applications. And we have center in many places of Brazil. And the result is very good. We've been able to improve it from traditional 83, 85% efficiency of this skin cancer to about 95 with this big program we have, treating many thousand lesions. And now we have a challenge here, which is reach 97, which is considered almost compatible with surgery. And to do that, we are implementing not nano, but micro needles, solvable micro needles to really increase the delivery of drug, increase the performance. We believe we will be reaching a wonderful 97% efficiency, which then the procedure and the technique can be adopted without any question for the public health in Brazil. And in fact, we are showing how the advantages of using soluble microneedles, which are needles that have the photosynthesizer on it, and we deliver it to the skin lesions by pressing it and making this solution of the needle inside the tissue. And we are proving that that can reach better depth of application of the PDT, as well as better uniformity. Because when you treat the planar lesions or skin lesions, you have to have a good uniformity of the photosynthesizer. Otherwise, you leave islands that you did not treat very well. Okay, so said this, and of course, cervical cancer is one of the targets we have, and we are also going the same direction of the skin cancer with the devices approved, with national team involved, and we hope we can bring the cervical cancer problem research with PDT at the same level that we are with the skin cancer, which is a big success in Brazil. Many people are adopting, people are doing that on a regular basis in many centers, including the National Institute of Cancer in Brazil, which declared that PDT for pre-cancer and small cancer lesion has changed their way of routinely treating these small lesions here in Brazil. Okay, well, condyloma is the beginning of the disinfection type and is also going very well. And now I'm going more towards the decontamination type of thing. And I mean, I already presented this many times in IPA meetings. which is the onychomycosis, which is something that nobody's going to die with that, but they die by taking a lot of fungicide to kill the fungus that's on the nail, on the knit of the nail, in this onychomycosis type. And PDT is a very good hope for this and for many other localized infections that can be treated. And I know that Vandana is a fan of many of the disinfection, with a wonderful work on the disinfection of the nose and everything. And this somehow adds together here. And of course, we are waiting for a nice meeting to present our work on the decontamination of organs for transplantation using PDT combined with the liquid that we circulate in the organ in a regular procedure, which is the perfusion. And we are doing this with Toronto with a big success, already papers published in Nature Communication and Lancet Journal of Medicine. And now we are really working hard to prepare machines that will allow a more broad use of decontamination of organs for transplantation. Here in Brazil, we are focusing on a kidney and liver decontamination for transplantation, and hope we can improve the availability and the success of transplant of organs using this wonderful technique of PDT and decontamination of the circulating liquid with UV light. They are both in the context of what IPA wants to do. Okay, but the main thing here is microbiological control, especially in the respiratory system, the infection. Before we had the pandemic of the present pandemic with the SARS and the COVID disease, we had a very nice paper published that says that 3.8 of global economy, 3.8 of global economy is a lot of money. It's 3.8% of all the money circulating around the world will be lost because of resistant bacteria. So we had a kind of pandemic being announced before with bacteria, and it's still going on. Bacteria are being resistant every day. And there is very little we can do with the present situation of the development of antibiotics. We all know that the companies declare that they need much more than a decade to reach the level that they need of new generations that will somehow solve this problem. Okay, and that's why we took that as a challenge as well, together with many people around the world, of course. And pneumonia is a kind of target, right? But it's very hard to treat pneumonia resistant. Normally the patient is there, sometimes intubated with endotracheal mechanical ventilation, and there's very little you can really do besides come with some engineering to really eliminate as much as you can without invades. We don't want to invade the body. We want to do everything extra body as much as possible. And that will be the focus of my talk. And as you can guess, there are many challenges in here. One is we have to illuminate from outside. So we have to study and guarantee that when we put the light from outside, they will reach the internal part of the lungs with the right intensity to be able to eliminate microorganisms, colonies there. A second thing, we have to worry about a very efficient delivery of a photosynthesizer, which in this case is being done by inhalation. So we came with this idea some time ago, already made animal models, and we are now facing the last part of it, which is really increasing the delivery of the photosynthesizer. And for that is where nanotechnology comes in. And that is where I will expend a little bit of time at the end of the talk, tell you what we're doing together with some groups around the world to increase the delivery of the drugs and being able to and being able to really do this in an efficient way. Okay, so first of all, concerning the resistant bacteria to antibiotic, many people around the world are doing many different types of work. And in those types of work, people are trying to show the same thing that we are showing here, that let's just leave a little bit the respiratory system away. And the question is, do you think that a tiny little PDT will potentialize the antibiotic, or maybe by a tiny PDT, we can revert the situation of resistant infections? And recently we have done a very nice work with Jennifer, which is a very nice PhD student we have in our lab. And we have been demonstrate that even though if you do a tiny little PDT, which is these red squares, see the red squares, they're not killing a lot. This is an experiment that you put together Staphylococcus aureus, and you do a PDT, subdoses PDT, a PDT that just damage a little bit. And that's why we have a very, the red squares are on the top, very tiny decreasing of the contamination with the PDT alone. And then we tested the effect in three different antibiotics, which is amoxicillin, erythromycin, and gentamicin, which are conventional antibiotics used, and that many bacteria is getting resistant. And what's happening is when we do that together, we do a little PDT followed by the antibiotic administration, this whole thing comes down. So a tiny little damage caused by the oxidation introduced by the photodynamic therapy can potentialize the antibiotic action. So as you can see, probably at the end of our research, we're going to combine things. We're going to deliver the antibiotics, deliver the photosynthesizer, do everything, and hope for the fully recover of the patients. But we want to talk about the respiratory tract, and that's involved more than just the lungs. We have the nasal part, the nose. Vandana is doing a wonderful work, and many other people on decontamination of that. We have the oral cavity, which is a very important inlet for bacteria to the lung. And then we have the trachea, right? And we have the larynx as well, because infections gets there. And then we go to the lungs and the bronchioles, and we have infection there. So I'm gonna try to show to you that we're going from the beginning to the end by doing PDT and demonstrating the efficiency. Okay, for the oral mouth decontamination, this is a work we have done many years ago. Just by a rinse of the mouth with some curcumin solutions, we can decrease considerably the amount of contaminants we have there. So this is a very good practice for like dental office that they are exposed to all the things there. I know that the contamination comes from inside as well, but if you can decontaminate the mouth by simple procedures, is a good thing. And as we show, by just using very tiny solution of curcumin, I'm a fan of curcumin, of course, and then a blue light, the way you see in the picture, we can decrease considerably the amount of bacteria on the mouth of the patients. So that contribute for whatever comes after. And we have been doing a very systematic study with different groups, showing that actually we can leave very little infection on the mouth. And then, of course, after 2 hours, this will recover if we have done a time following up of this. But what's interesting that even with 1 gram per liter of curcumin, which is not too much, you can reach a wonderful without having any color of the mouth or the teeth. And we have, as I told you, by doing time following up, we've been seeing how long this stays there, how long it takes for the recolonization, and we have done that as well. Okay. And we have done a model for make some considerations. I'm a physicist, so physicists like to do models. By considering that the photosynthesizer will reach the microorganism in a random way. We were able to do a model that can predict what kind of concentration you need to use and what kind of light doses you need to give to reach some survival. Of course, we want zero survival. We want to eliminate everything. So you need sometimes large concentration and large doses. And we test the model with many different microorganisms to show that actually this is a simple and elegant model because it can predict different levels of elimination of microorganisms. Because sometimes you want to decrease without fully eliminating the microorganism because you have some good microorganisms. Okay, so this is a nice model and I've been applied for many years now to like start the planning of PDT section for decontamination. But, sorry. We're gonna talk about the first part of the infection. I'm not gonna talk about the nose because I don't have work on that. And but if you go to the internet, Vandana and many people are doing wonderful work. But I'm gonna talk about these pharyngotonsillitis, which is a main problem for antibiotics because any kid has to experience a few antibiotics every year, and that's increasing the possibility for developing the resistance of the bacteria. And the idea here is very simple. If we can make the drug to reach the infection, like you can see, and then we illuminate, we're gonna be able to reduce considerably the infection. This is pharyngotonsillitis is a big problem everywhere in the world and some countries. You see here the number of people that has to be treated. The incidence is fantastic, right? It's enormous. Is a really this data is before the pandemic. Now we don't know because many people are trying to treat their throat infection at home to avoid going to hospitals or anything. So we don't know exactly. But even though before was a big deal, and a big deal which consumes antibiotics a lot and sometimes do not solve the problem because it's being harder and harder. I know many people that have a kind of chronic pharyngotonsillitis infection, and that has to be treated with alternative ways. Antibiotics are getting resistant, and there are many side effects if you abuse. So we came with this procedure that we do a fluorescence examination, make collection of the microorganisms, give a kind of a pill or syrup with a spoon, or a pop when he has a young kid, a lollipop, and that's dissolving like 5 minutes, going to impregnate the photosynthesizer on those infection sites, and then we do the treatment. It's very interesting because it took us about 2 years to get the the ethics commission permission to do the experiments that we are doing now, and that is going in. We use curcumin as well, and you see we use so much curcumin that at the end I'm going to show to you that now we are synthesizing curcumin to make everything lower price. We have a device. The device was approved by authorities here, but the drug is not approved yet. So, because this is a combination of fluorescence to follow up and to see the infection, followed by the drug that's administered orally and the illumination device. And we've been doing many different works along the years, starting from in vitro tests by collecting the bacteria of the infections, or even virals we're doing, as you're going to see at the end, and make the application and see the advantages of the PDT. Okay, in the experiments that we have done collecting bacteria, we see that the PDT with curcumin is very good in eliminating almost all types of bacteria that can be in the pharyngotonsillitis type of infection. And this is being done, and we see that you can reach about something like 5 log scale decreasing on the infection. How is the procedure? You do the collection before and after doing the PDT. And then there is even some follow-up. And then we regulate what kind of light doses we should be able to use on the throat. And that's controlled by the time of illumination. Okay, so as you can see, by looking, we're using local fluorescence. We see the infection, we do the PDT. Very short after that, seems that the activity of the infection that we look by the red fluorescence that's given by the porphyrin produced by the microorganism is over. So means that we have a fast response of the PDT treatment for those infections. We have done that in two different groups of people, adults and young kids. Young means not baby, but like between 3 and 10 years old. And the way to administrate the photosynthesizer is being done by either liquid when are adults or a little candy that people keep on the mouth and that give you the right time for the incubation, or then the lollipop, which is very popular for young kids. Okay, for adults, we have done a quite large number of adults, 98 cases were treated, and 92% they got rid of all the signs of infection in less than 48 hours, right? And you know, antibiotics will take 48 hours to start to act. So here we see the absence of symptoms, and we make collection in after 24 hours and 48 hours. And in 80% was resistant to PDT, or PDT was not well applied because we have control, but not perfectly on the uptaking of the drug by the colonies that's localized on the pharynx. For young kids, we got some similar results. And what was interesting, this is after 24 hours, the symptoms run away. And this is no application of antibiotics together. Now we're doing a full clinical trial that we do the antibiotics together with PDT and follow up. So because of the systemic fever that can have an infection and everything. So we were approved to do that. Now we are starting to increase the number with more doctors participating. But even though, you know, in 94% of the cases, you can deal with this infection by doing PDT. And of course, if the things persist, you should go for the traditional treatment. And we now have a permission to do a big clinical trial. So we were in 2 different hospitals. We even made a small ambulatory to get the adults, but then came the pandemic. We have to stop because it was in the same hospital that's the main headquarters for the COVID people here in Brazil, or in São Carlos where I live. And the idea was collection, following up for a long time. And we were able to test the content of a viral type of infection and bacteria. And what's interesting is that we did antibiotic in all the patients, but for some of them, we did a placebo PDT. And we were able to follow up the people that we did and the people that we did not. And this very nice plot, which we call cumulative survival symptoms, is how the patients progress after the PDT. This is a single PDT section, okay? But they took antibiotic. But what's interesting is that if they show any symptom, we consider still sick. And we only start to count the people that got out of the disease when no symptom is showing. And as you can see, the people that really took the PDT, it comes much faster down than the people that only took the antibiotic. The antibiotic will reach the PDT level at the very end after many days of after the treatment. And then we come with this index that shows the superiority of having PDT. If you go to the end, if you go like 30 days after the treatment, and then you see how much PDT is better than what you're getting. So that's a function of time. And as you can see, the superiority, it grows as the time passes by. So there is a very nice long-term effect when you do the PDT for those pharyngotonsillites in adults. And now we select from a big group only the people with the virus infection. We know that antibiotic doesn't do anything there. But for the people that took the PDT, the infection came down much fast. As you can see, in 3 days, we get rid of the, start to decrease considerably the people that has a viral infection, not bacterial, by virus, compare with the PDT and the PDT placebo. So it seems to be very good even for the infections that are based on virus. Okay, now let's go a little down. We were in the larynx, let's go to the trachea. We all know that the number of people that needs the ventilator, the mechanical ventilator is big now, especially in the COVID, but even before and that's when we put that tube inside the trachea is an opportunity for nucleation of colonies of bacteria. And that's why when you are intubated, you have to go together receiving antibiotics to prevent infection. So the surface of the tube does not get any antibiotic. There is no circulation there. So there the infection persists. So you try to treat everything else, but the colonies of bacteria may stay there. And normally you make an intubation, you put the tube in the patient, and that persists for some time. So the idea is very simple. Let's put photosynthesizer on the tube and illuminate the tube as the patient is intubated with a fiber that goes inside the endotracheal tube, and hope that will produce a nice effect in preventing infection. In fact, the idea is, you know, the film, the biofilm grows inside the surface of the tube, and the idea is we functionalize the endotracheal tube and made a device which is a tiny little fiber that diffuses light inside. Okay, so the way to do this is by making a very nice chemistry with we have the help of the group from Coimbra and even from Lucas, which is the people, and Amanda that are doing the functionalization of the tubes. And then you have a whole different way of making sure the photosynthesizer sticks to the surface and it stays there, right? It cannot be washed out. It has to stay there acting for a long time. And we have been doing all types of study. The first thing that everybody asks is, are you changing the property of the endotracheal tube by doing this functionalization? And the answer is no. We are keeping all the properties of the tube. And of course, we make all kinds of investigation showing that everything is uniform, covered. So we are able to do a good job doing photodynamic on the colonies that stick, trying to make a biofilm in this tube. And the response was very good. We have done a wonderful work in vitro, and now we are preparing to do it in animals. And the result is very good. We can basically eliminate the formation of biofilms. That's a very nice plot, which I like very much. Is the the tube, and you see the evolution of the growing of the microorganism at the surface when you're not doing PDT or when you're doing PDT. When you're doing PDT, you basically stop the formation of the colonies and the progression of the infection due to this endotracheal tube. And now to evolve with this, we'll be doing a nice artificial trachea to be able to study all of this. This is a wonderful device. We hope to progress a lot by doing all of this before going to the patients. We want to go to pigs. We are making now all the program to do the first pig type of experiments. And this is very nice, it's secured because we only kill what is attached to the tube, which is the main focus of the infection. And the light on the trachea doesn't do anything because there is no large concentration of photosynthesizer there. But that's bring me to the main topic, which is the treatment of the pneumonia. And The idea also is very simple. We only work with simple things. The idea is inhalate a photosynthesizer, and we're gonna do an extra corporeal illumination, reaching the lungs with infrared light, of course, and try to reduce the infection. The first models were done, we are always using in the green and the cyanide, ICG, right? Because this has a very nice band absorption in infrared, and they are very efficient with those kind of microorganisms. The first experiment we did is already published. We did it with small animals and induced the pneumonia in those animals. And the ones we treat by making the inhalation and the illumination, we make a full elimination of pneumonia. And the ones that we did, you show the decrease here in the survival plot in this Kleppmeier type of surviving curve. So there is a big chance that this works for big animals, and we are doing for that. And of course, there are many things we are worried about in this procedure: security, not hurting the normal cells, and distribution of the photosynthesizer. And many work is being done on this. And we want also to try to evaluate the photoreaction going on. So we want to understand very well how the bleaching takes place to be able to follow the photoreaction and the photodynamic action going on there. We were doing first in 780 nanometers, but now we jump to 880 nanometers, which seems to be even more efficient, and many different type of work is being done here. We are always doing in vitro efficiency for everything we do, in vitro safety and then animal safety, using mouse model in your laboratories. And so far, the experiment is going very well in nebulizing, transferring the photosynthesizer to the lungs, and doing the treatment and eliminating the pneumonia. Of course, we want to do this in people because this is the big demand. But before going to people, we're going to have to go for big animals. And we are making a whole set of experiments, already started experiment with pigs. And we want to first to show the efficiency of the process in pigs. And then we're going to go to the people, we hope. And I understand that at the moment, this will be a good addition to helping treating people in the hospitals and everything. And we are even thinking about to see if the elimination of virus due to the COVID can be done by extra body illumination and the breathing of the photosynthesizer. But we did not reach there. We are in the in vitro experiment with the virus yet. We do all kinds of understanding of the basics of what's going on to be able to reach, of course, the final goal, which is going to the people. And, you know, first of all, we are very worried about light. Then we show that we can really organize to reach the level of light we need to kill the bacteria very efficient when we pass the light to a big barrier. So we made what we call experiments using phantoms, and the microorganisms underneath of the phantoms or in the top, depending where you illuminate. And we're seeing those plots, which is the percentage of that. And then you see that controlling the time and the amount of light, controlling the amount of light you deliver, controlling the time of illumination, we'll be able to always take to complete elimination of the bacteria in that case, even with very thick layers that simulate the thorax walls, right? And we're doing now in pigs. And the first experiment that was done in pigs was to analyze the inner part of the thorax chamber, using foresee the light distribution and seeing seeds, putting seeds of bacteria that will be reached by the light and eliminate. And as you can see here, we see the decrease when we have only light or when we have only drug. This is always the control. But then in different positions of the thorax chamber, we were able to show that everywhere in the thorax chamber of the pig, we are able to decrease to zero, basically, the amount of microorganisms even illuminating from one side. Of course, our plan is to eliminate both sides. So you're going to have a front and back or right and left illumination of the lungs. And these devices has been done already, and we are in final tests using either pulsed light or continuous light. And this has been a wonderful experiment. And as you can see, there are very nice things in here. Because if you look at position 4 related to position 3, you double the distance from the light source. And then in any regular things, you're going to expect this to come into the 1/4 of the efficiency, because the light, when propagates, diffuses to 1/4 distance square. But no, it decreases only a small factor. Actually, decreases only about a few factors compared to what we expected. And the reason for that is that thanks to the thorax geometry, when you send light to the wall, external wall, the light inside, as you could see in this picture here that shows the inner part of the thorax chamber of a pig, the illumination goes all the walls diffuse light as if it was a small diffusing source, isotropic diffusing source. And we know that the intensity in this case drops with the distance square. But because the light is coming from many places, when you add up inside the thorax chamber, it's like a constant illumination. In that case, it was not the center and the walls did not get the same result because we had only one side. But when you illuminate from both sides, the inner part of the thorax chamber will be like a constant, almost constant illumination of light, as can be proved by the equations. And that's a very good thing, because we know that the lungs is almost, it's very transparent to this light. So if we make the light uniformly go, we uniformly going to illuminate the organ and eliminate the infections that are there. We're doing many experiments that can prove those ideas and the efficiency we're going to have when we want to treat the real pneumonia in people. But the big problem here is delivering the drug. And to do that comes the nano-emulsion. And I have a very nice researcher here, which is collaborating with Canada, and we are doing a very nice nanoparticles for PDT using those liposomes. And there are many ways to put the drug in those particles, as you can see in this transparency. And using the liposomes, we can put porphyrin, we can put ICG, and we have a uniform distribution, which means that those particles can go deep in the lungs and then release the photosynthesizer. We don't leave a lot of sensitizer on the way in, which is what we want. We don't want to photosynthesize everything. We just want to photosynthesize the air tract, right? Where are the colonies that are preventing people to breathe? And then this work with the liposomes, right? This is basically what we're doing. These nano-emulsions, is very good. And we are doing very well in treating with those. There are big advantages on that. And we even thinking now in ways it's very stable. It can go and then liberate. Will not liberate things before or degrade very fast. So we can go actually to the target, which is the lungs, make the treatment and not putting a lot of drug on the circulation of the patients. So that is a very nice window of time where we liberate the drug is still on the air tract, it is sensitizing the colonies of pneumonia, of the bacteria, and we do the treatment. And we are doing all kinds of study to make sure that we going to understand what's going on, how is the aggregates of those particles, and how they liberate inside the lungs to make sure we have the things under control. Well, I could talk a little bit about food, which is a big problem, right? We want to clean food as well. And we're doing that also with curcumin, which is wonderful. Now, curcumin is quite expensive, you know, and then we made a whole project together with a company to be able to produce in big quantities, synthesize, based on a natural extract. We make a synthesis of curcumin in very big quantities. So now we are able to produce many kilos, and that make it possible to use it for those in a very inexpensive way, because normally photosynthesizer, they are expensive. And except methylene blue, the other ones are quite expensive. And for the kind of treatments we are developing, we need curcumin. And we need the curcumin very pure. We don't want to do natural extract that comes with the balance between the three types of curcuminoids. And depending on the region that you took those vegetables, we want to do the synthesis, and this is going very well. It's already being transferred to the company, and we hope to be able to produce curcumin inexpensive enough to do this kind of treatment. And we are going now for producing in big quantities as well, using chemical technology, ALA, methyl ALA, and now the ICG. We will start very soon, because if we're going to do the treatment of the lungs with ICG, we want to make sure we can produce in large quantity and not pay a fortune per milligram as it is today. Well, I come to the end. What I want to give you in this nice seminar of the IPA is an overall view of what we're doing for the decontamination of the respiratory system, which is one of the challenges we have now concerning the action of antibiotics. And, you know, as I said, after the storm of the COVID, we're going to have a lot of people with effects left behind on the respiratory system because of the infection. And they will be very susceptible to infections. And we have to, maybe techniques like this will help. And this is our main goal. Our main aim in here is to contribute to the society in that way. Thank you very much. And if you have questions, I will be happy to try to answer. Otherwise, I will get help of my team that, of course, they can help. They can answer those questions. Thank you.
Thank you so much, Vanderlei, for this really nice talk. So please, if you have any questions, think about putting them in the Q&A window. We actually have already a few questions that came during the talk. So we have a question about how the nanoparticles, the photosynthesizer, sorry, leak from the nanoparticles. So somebody's asking, have you studied the leaking of the photosynthesizer from the nanoparticles? Okay, well, this is actually, this is a nano emulsion. It's made with an oil type. And the photosynthesizer, the present time, the way we can put inside, but normally we are doing outside. And when go to the lung, it's liberate, because naturally we'll find an environment that's appropriate to liberation. And we are also trying to do some ways of liberate from outside as well, using different techniques that we are now investigating to do that, using some mechanical effects. So it liberates, and of course there is a time of liberation, and normally we are studying this to make sure that we wait to the right time to have the right sensitization to do the treatment. Okay, thank you. So we have another question that come from Mafalda. So she's asking, in your study, increasing the effect of, so in the study where you show that the effect of PDT enhanced the effect of the antibiotics, which sensitizer were you using? Well, we use, I think for that specific experiment, we use a lot of sensitizers. They always, you know, we try to get the best that we have. But I think for that experiment, we're using the curcumin as a sensitizer. But, you know, porphyrin, chlorine, radiochlorine, almost all sensitizers are good in some sense for killing microorganisms. The thing is, some of them is easier to work,
And since we are very much interested in going deep in the body, we needed to worry about how we're going to excite. So that sometimes limited what kind of photosynthesizer we're going to use.
Yeah. Okay, great.
But the question is, you know, what's important is that works also for resistant bacteria. So resistant bacteria lose their resistance a little bit when you're doing a previous PDT. And that's why it's interesting for throat infection and infections that you can go first do a PDT section and then go on with the thing.
Okay, thank you. So there is another question. I'm going to read it because it's quite long. It's from Mons. So the question is, many researchers working on PDI antimicrobial PDT mentioned that the microbes do not become resistant to PDT. However, human cells can have an antioxidant stress response, helping them to cope with the effects of PDT. Do such stress pathways not exist in bacteria and other microbes, or are repeated PDT treatments equally effective?
Well, look, we have tested the development of resistance to PDT in vitro. We never really made the animal models and things like this. So far, it's been okay. We didn't see big resistant development. But the question is very legitimate, and we're gonna have to go in more complex environment. That's true. We have to go to more complex environment to see. But normally, there is a work we have done some time ago to show that if there is a kind of sequential treatment that has to be done, and if you are selecting by threshold, you are leaving a little bit behind the microorganism that could not be killed, you just do a compensation by increasing a little bit in the next treatment, the drug concentration as well as the light dose. Of course, there is a limit where we can go. But my idea is that in 1 or 2 or 3 treatments, we somehow eliminate the infection. So we're not gonna give a lot of chances for the microorganism to develop a lot of resistance. But I agree, we should do more animal infection to see if they get resistant. In a few sections we didn't see, and in vitro, normally we don't see a severe development of any type of, like we see in antibiotics, right? Antibiotics, a few generations is already, you have to double the dose of antibiotics to be able to kill. We don't see that in PDT, but there are many work and many people are doing that to clarify. And your suggestion is very good. I'm gonna take note that we have to do in the in vivo environment if we want to make any statement that the people will pay attention. Thank you.
Thank you. Okay, I'm going to take one last question. So the question is, I heard ICG has low singlet oxygen yield. Why do you think it works so well in the animal pneumonia model? And the second question is, how does curcumin compare to methylene blue for all and throat clinical PDT?
Okay, you see, I have to ask you to repeat the question.
Yeah, sure. Yeah,
I'm trying to read here the question.
yeah, it's not in the same.
It's not the same. Yeah, that's what I realized. Okay, well, can you just repeat very fast? I mean, the ICG, why is doing well?
Yeah, so the question is that ICG has low singlet oxygen yield. So why do you think it works so well in the animal pneumonia model?
Well, I mean, it's low, but it's enough to produce the damage you need to eliminate the infection. Everything is a compensation of those, you know, for how long I'm gonna do, and hopefully the molecules do not destroy themselves. Sometimes not having a very big quantum yield is not so bad, because you make one molecule to generate many oxygen without dying by their own product, by bleaching. So far, the result shows that it's quite efficient. And we've been using, and it seems to be a good way to go. Of course, other type of photosynthesizers may be a big advantage, but I always think that we have to make the best use of one photosynthesizer before jump to the other. When photosynthesizer became the only limitation, then we jump.
Okay, great, thank you. So sorry for the question that we didn't answer. We're gonna go to our next speaker. So Pilar, I give you the microphone again.
Thank you, Ana Laura, and thank you, Vanderlei, for the nice talk. Very interesting. And yeah, we move now to the third speaker of the session, our last speaker. Professor Jonathan Lovell is